I may actually grab a chair because I have to do a bunch of typing here. Can everybody hear me okay? All right. Okay, so I'm just going to walk you guys through the process of generating the metadata templates and spreadsheets, how those get filled out, how you upload metadata and sequence data to the website.
Doug, you're blocking the connection on the local laptop.
Yeah. So as Rob mentioned, excuse me, you can sign up for an account on here if you don't already have one. This is an automated process, so if you don't have one, you can just click on the Create Account link. It'll just ask for some very basic information, send you an email confirmation to which you have to, it just sends you a confirmation link. So you click on that and it will validate your account, and then you'll be able to log in. Okay. So when you first log in, as you've seen, there's just a few basic options available to you. You can perform a meta-analysis, which Rob has already walked through, so I won't really get into any additional details there. But what I am going to talk about is the process of creating a new study and filling all that stuff out and doing the basic processing so that you can get to doing the actual analysis of your data. So I'm just going to go ahead and create a new study here. We'll just give it some test name here. Obviously, you should be filling out much more meaningful values than I'm going to. Anything that's marked with an asterisk here is actually required, and anything else that's not, you can leave blank, but if you have the information, it's always better to put it in. If you have a PubMed ID already associated to your study, or after you publish it, you get one, you can enter it here, and when the studies are viewable on the website, there will actually be a clickable link that will take you right to the article, which is nice. And these are all of the different types of studies that are defined in the mixed standard. I'm just going to leave it on MyMarks survey, which is what my test data set is. So I have some test data, a very small data set that's actually a soil study, so I'm just going to pick that. But you can actually select as many of these different package types as you want. And these are all the package types that are defined in the MyMarks standard, or the mixed standard, I should say. Yeah,
when is the built environment package going to show up there?
I actually have almost all of it scripted out, so it'll probably be maybe a week or something like that. I just didn't want to introduce a brand new package before the seminar started.
Doug,
yeah.
Can you increase the font size?
Oh yeah. How's that?
Better.
Better? As Rob also mentioned, so the built environment spreadsheet was released, or at least a copy of it was put out probably, I don't know, maybe 6 to 9 months ago or something like that. It looks like it probably needs a little bit of work just in terms of the controlled vocabularies that are in there. They're fairly sparse at the moment, but the basics are there. So anyway, let's see, we'll put in something here. Once I filled everything out that I have, or that I actually need to fill out, I'm just going to click Create Study, and you'll see that we have a few more options available to us once I do this. So we have, in addition to all the stuff that we had before, under the Manage Study, now we have a whole bunch of other stuff. We can generate MyMarks compliant metadata templates, which I'll walk you through in a minute. We can add sequence files to the study, and we can add metadata to the study. So the first thing you need to do is generate your MyMarks compliant metadata templates. So the screen that you get when you do this, it looks a little bit busy, but it's really pretty simple. At the top of the screen, it's going to ask you how many samples you have in your study. The reason we have this up here now is that it will actually do its best to pre-fill your metadata templates for you, so it can save you a lot of work if you just put in a little time up front. So I'm just going to say I have 20 samples. And then you can see the general organization here. The gray bars categorize all of the different stuff. So we have sample fields at the top. Everything that's in red is required. Anything that's in gray is an optional field, and these always have a checkbox next to them. So if you do not check it, they will not show up in your templates at the end. So we'll just go ahead and check it. So we have sample fields at the very top. We have library prep / sequence fields down here. And this is all the stuff that's necessary for actually doing your PCR and you know, all the pre-work that you do to get it into the sequencing hardware. And then underneath here, so if you remember, I picked both soil and sediment packages when I created the study. And so what happens is that it goes out and figures out what all of the specific fields are for the study types that you picked, and then it just enumerates all of those down here. And almost always in the mixed standard, the package-specific fields are optional. It lists out the things that are possible, and then you just pick the things that are actually relevant to your study. So you can see they're all alphabetized in here. And anything you actually want, you just tick it off. And if you have something you'd like to populate it with, you can type it in. If you don't know exactly what it is, or you know it's going to be different for every sample, you can leave it blank and then fill it out when you actually get your templates.
The units are there somewhere?
Most of the fields that have a measurement in them, like distance, it will say in the description what the units are supposed to be. And in cases like Rob mentioned for age, it will actually have a unit selection. And then when you submit it, it will do an automatic conversion for you from...
On this interface, I notice it doesn't have a unit shown as a default. Is that something you guys could add so you don't have to have a separate sheet?
Generally, in the description itself, it will say what the units are supposed to be.
No, I understand that, but is the units in a set, I'm sorry, is this description in a separate set of fields that then you dig through that Excel spreadsheet, look down, or?
I will show you in a minute.
Okay.
So we do produce, when you select the fields here, so anything that's required and anything that you selected will actually show up as an additional documentation file that you get when you download your templates. So you can always refer to that to see exactly what the units are supposed to be. And then when you fill it out and upload it, it will do its best.
This is just building what
This is
you want
just,
to put it in. It's not
exactly.
really putting in all your values.
That is correct.
Okay.
You can pre-fill a lot of this stuff, but this is just generating templates that then you are responsible for filling out. Oh, yeah. Good question, though. So we can fill out stuff here. For example, I can put in some taxon ID. We can give our samples a default title if we want, or we can leave it blank and fill it out later. I'll just fill a couple of these things out so we can see what happens when I actually submit this. Some of the fields, if it's an ontology, we were trying to save you a little bit of work here. So you can see that this is actually going out to the bioportal directly and querying the ontology, so you don't have to know exactly what the values are, you can just go look for them. And if you do it this way, I guarantee you that it will pass the validator, so you don't have to deal with all of the ontology lookups. This will save you a lot of time. Same thing with anything that says data type is ontology, they're all hooked up the same way. So let's say this is a tundra, I don't know, lake bed. Oh, and the other nice thing is that even if the term you selected isn't an actual term, if there's a synonym for it, it will show up. So in this case, the acceptable term is dry lake, so we can just pick that. Let's say it's mud. It'll give you all the options in the ontology that are relevant to that particular term. So this is a great way to save yourself a whole bunch of time.
Do you have a bunch of samples where most of the information is the same?
Yep. That's what this is for. I'll show you in just a second. So if you remember at the very top I said I have 20 samples.
Got it. So you could
It's going to
carry it
pre-fill
over
the
or
sheet,
whatever.
exactly. And so if you know you have things that are going to vary from sample to sample that you don't want to fill out, then you just leave them blank. And it'll still create the 20 rows and fill out everything that it can, and then just let you fill out the rest.
How about you create a field that is not there?
I'll do that for you. I'll show you what happens. Okay, so the stuff at the top is the sample template. The stuff at the bottom here is the prep template. I'm not going to fill out too much here. I'll put something in here just so you can see what happens when we fill these things out. So that's all I'm going to pick for now. Just going to leave everything that's optional blank. The other thing that we have is there's a lot of fields in the standard, and like I said before, we picked a soil study and a sediment study, and so those are the fields we see by default, but there's actually a lot of other fields in the standard that we might want to add to our study. So if you click in this box at the bottom, by default it's going to go look for all of the other fields that aren't in the list above, and you can start typing in here. Let's say we know we have something with carbon. So we can just type carbon in here, and it will go find all the fields in which the title has carbon or the description has carbon in it, and we can add those to our study, and they'll show up in the template, so you can see that they show up above here. So let's say we're satisfied with that. I'm just going to click Generate Templates. And what you get is a zip archive of all of the documents. You can view them on here. Obviously, this isn't particularly useful, but if you just wanted to do a sanity check, you could. And this is the documentation file I was talking about, Carrie. So when you generate your templates, anything that you picked and anything that is mandatory is going to show up in here. It'll tell you what the data type is, generally what the expected values are, and some description of it. And if it's something like, you know,
The value should be, is that the right is my meters? When we look at the description, if we go to the right, is that where it's?
yeah, sorry, it's just,
Okay,
yeah,
got it.
it does say it in there.
Okay.
But this is the reference document, and when we download this archive, you can see that those 3 files are in here. So you always have this field reference document available to you. And these are the files that we pre-generated. So if I open this in Excel, for example, you'll see that we have the 20 rows that we said we wanted to generate, and anything that we had pre-filled, it will just pre-fill for you. So we've saved ourselves a little bit of trouble. We could have done more work up front and had very little to do in these templates. So that's nice. That saves you a whole bunch of time. The prep template is exactly the same way. We only picked one field, so it pre-filled one field for us. So those are the files that are generated. But for the sake of the demo, what I'm going to do is just go ahead and use some pre-filled templates so we don't have to go through the process of actually making that. All right, so what I'm going to do, so we've generated our templates, so we've done the first part. Now what we need to do is upload some sequence data and also some completed metadata to our study. So I'll show you how to do that. We'll go ahead and do the sequence files first. So this is an Illumina study, so we have a FASTQ file and a barc. There's a sequences file and a barcodes file, both of which are FASTQ files. They're required to be zipped just for size reasons, and so both of these are gzipped files. The easiest way to do, I'll show you what this looks like here. There's a little shortcut I just discovered recently that's sort of nice. You can click Add and browse for all of these files, but it's somewhat painful. You can also just copy these to the clipboard and paste them into the uploader, which is much easier, so we're just going to do that. All right. These are very small files, so that was very quick. And you can see what files you've uploaded to your study once they're up there. We have the barcodes file and we have a sequences file. We have the option of removing these from our study if we somehow uploaded the wrong things, then we can fix that. The next thing we're going to do is go ahead and upload the metadata. Same process here. I have a real short set of metadata just to keep things a little bit quicker. So I'm going to copy that and paste that into the uploader. And so what happens when you do this, there's a whole bunch of stuff going on behind the scenes here. All of that stuff that just flashed by is the validator checking to make sure that everything in those templates is actually okay. So it's going through all the rules that are defined in the MyMark standard and making sure that those are valid.
One thing I should point out, though, was that it's really easy to have a file that meets the standard, but it's semantically incorrect. And that's the reason why Gail and other people in the group put in a whole lot of effort in checking those manually before they're uploaded into the database. So that's how we get things like making sure the ontology annotations are consistent, making sure the parameters are recorded in a consistent way, and that kind of thing. So it's one of these things where probably no automated system will ever really be able to do it. You need to have a person looking at it and figuring out whether it makes sense, as well as being technically compliant with the standard. Yep.
So if everything goes well, you'll get fireworks at the bottom. It's your happy reward. We can go back to the study, and we should see that we have both sequence data uploaded and we have metadata uploaded. And once that happens, we can now actually submit the job to QIIME for processing. And what happens in this processing job is it demultiplexes all of the files that you've uploaded. If necessary, it will perform possibly, depending on what it is, possibly some trimming and some minimal quality filtering. And it also does OTU selection, and this is all based on 97% match against the green genes tree.
Hey,
Oh,
Jared.
goodness.
Jared,
Yeah?
yeah, you should tell them that they don't get to submit the job yet.
All right.
They get a message that says Gail will submit it for them.
Yeah, we'll probably actually change that, but
Be nice to Gail.
yeah. So just to keep things sane, we still do a review of the stuff that we get, and then once we're satisfied with what's actually up there, we will click Submit Job to QIIME. And so what happens at this point is it submits a new job, demultiplexes, does OTU selection, and once that's complete, we also verify that the output of those files actually looks reasonable. So for example, if somebody put in the wrong barcodes or the wrong linker primers or something like that, what you'll wind up with is very low or no hits in your OTU selection and also from demultiplexing. So we verify that those actually look sane, and if they do look sane, we'll upload those to the database. So the job is just running in the background now, and once it's done, we'll have the option to actually load all of those OTUs and sequences into the database. And once that whole process is complete, that study becomes available for a meta-analysis, which is what Rob has already covered. So the one request was that I show you how to add additional fields that don't exist in the standard at all. So we'll just go ahead and do that. So what I'm going to do is just go ahead and open my sample template in Excel.
Could you also stick in some incorrect values in some of the required
Yeah,
fields to
definitely.
get an idea of what that interface looks like?
Yes. So we'll just say public is 234, and we'll add some brand new column called, I don't know, brand new column. Okay, we'll go ahead and save that. If you're adding your own data, you could add 2 columns to it if you wanted to. For example, if you're introducing some brand new measurement that nobody has used before, you would probably want to introduce 2 columns. One would be the name of the measurement, and the other would be the units.
Okay.
Or what you would do is what we have done
And then
is,
the value would go third and name, value, yeah.
or yeah, exactly. Or what you can do is introduce a single column, and then you actually have an opportunity to add a description to this when it gets uploaded. And you can say should be in meters, should be in grams, or whatever.
So you can do that, all right.
Yeah.
That seems easier than having 3 different columns, right?
Which is why we have done that for most of the columns, because converting from everything to everything else is incredibly time-consuming and costly. Okay, so now we have a brand new metadata archive. Hopefully this won't die. We'll try this. So all I did there was just go ahead and I edited my file, and I just zipped up both the sample template and the prep template into a new archive, which is what's required by the uploader. And I'm just going to paste that in, and we'll see what happens here. So here we go. This is what happens when you introduce something that the database has never seen before. So here it is. It's called My New Column. I can give it some description, like you said, should be in grams or something like that, if that's how you want to handle the units. And then you can give it a data type. So anything is just a text value. You can type in whatever you like. It can be a number range, or a date. And so these are what the validators would pick up on. We'll just leave it as anything for now. So now it's going to go ahead and validate the prep template's okay, but I did make a change in the sample template. And so this is what happens if you have errors. It will show you the row that has a problem, and then you can just scroll through and see which column actually had some issue as well. So 234 is not a legitimate value for public, it's a little cramped right now. It looks a little bit nicer when it's spread out. But if you click in here, it will actually tell you what the field name is, what the data type is supposed to be, what the expected values are, and some details about the column. And so you realize, oh, that was a mistake. I should just type in something else. And if you type in something that's legitimate, it will turn green and actually let you submit the metadata to the database. And we go through this whole process again. And once
and you probably saw the little update all like items link. So if there's a whole lot of the same incorrect value, you only have to fix it once, and then all the other ones will also commit.
Yeah. And there we go. So that uploaded just fine. So we have one brand new column associated to this study. And if anybody ever uses that column name again, what happens is that the database recognizes it's in there, and it will present to the user the fact that this column already exists. It's not part of the standard, but it exists, and it will tell you what it is, what the data type is, and ask if you want to use it. Because perhaps you have something in your study that's like a similar concept, but it's you want to store it differently or you need to deal with it in a different way. So it'll tell you what's in there and what the units are, what the data type is. And if you need to change that, then you just change it in your templates and re-upload it so that you don't wind up with a conflict of an existing field. But if you do want to use it, that's actually great because you actually can compare that field to another study that happened to use that field as well. So that's actually really nice. Yeah, so that's pretty much it. Does anybody have any questions about the metadata portion?
Kind of a related question. So like, if we have like a FASTQ file that has samples from multiple studies or samples that aren't really included in that study, would you rather it be parsed to only include the barcodes in that study, or rather upload the whole thing and only include in the barcode file the relevant samples?
What most people do is just send us the entire file, and then we do the demultiplexing and only store those for which you provided barcodes. But you could, if you wanted to, demultiplex it yourself and send us whatever was actually relevant. It's a little bit more work, obviously, and what usually happens is that you get a per library file for every sample. But it's possible to do.
Doug's question.
Oh.
Do experimentally treated samples go in there? How does that come to that?
We definitely get metadata like that.
Can you repeat that question?
Oh, the question is, do experimental, essentially like controls or things like that, right?
Right. Did you make a distinction between? You should be able to make a distinction between controls versus treated, say, soil samples.
Right. Yeah, we do.
So people making comparisons across geographic regions, you would want to necessarily include experimental treatments.
Exactly, and so most of the time you just have to be careful how you annotate those samples so that when you do if you're doing a meta-analysis and you're selecting all the samples for, like they're all soil samples or something like that, you might want to actually pay attention to where those samples are coming from. There are cases where we leave some of those out if they contain basically no metadata whatsoever. You can still do an analysis on that with QIIME, but it may or may not be relevant for the database per se. But usually we collect them and store them, and then if you see anomalies when you do your analysis, make sure that you know what the source of those samples is. All right, well, thanks everyone.